The year is 400 BC. Although Hippocrates is not the first scholar to contribute to genetics, he did theorize the brick-and-mortar theory of genetics, which states that whatever traits a father has in all parts of his body, he will pass on to the child. Aristotle directly challenged the brick-and-mortar theory in 350 BC. He believed in direct inheritance of traits, meaning that parents pass their traits to their offspring through reproductive material, their gametes. He challenged the brick-and-mortar theory based on observation. If it was true, it would account for factors like age, voice, hair, nails, and so on. In the 19th century, a monk named Gregor Mendel carried out an experiment with pea plants, in which he sought to investigate patterns of inheritance. He did this using pea plants that varied in height, flower color, and seed shape. For example, when breeding a tall dominant plant with a short recessive plant, they all showed the dominant trait. When the first generation self-fertilized, 3 out of 4 plants showed the dominant trait and 1 plant showed the recessive trait. This was just one of the many parts of his experiment, but displays the advancement in scientific discovery. In his quest, he discovered the fundamental laws of inheritance as well as concluded that genes come in pairs from both parents. He formed 3 laws of inheritance: the law of dominance, law of segregation, and law of independent assortment. The law of dominance states that if a dominant allele is present, it will be expressed. The law of segregation states that homologous pairs split during anaphase 1, when meiosis takes place. Lastly, the law of independent assortment states that the orientation of one homologous pair does not affect the orientation of other homologous pairs, hence the name independent assortment. In the 19th century, Karl Krenz, Hugo de Vries, and Erich van Schermarck rediscovered Mendel's work independently. They did this separately using different plant hybrids. Their experiments helped to expand awareness of Mendel's laws of inheritance in the scientific community. They confirmed his discoveries and used their own work to support his laws. In 1928, Frederick Griffith carried out an experiment that suggested bacteria are capable of transferring genetic information through a process known as transformation. In his experiment, he used mice and different strains of bacteria. Griffith used 2 strains of the same bacteria that infect mice, a type 3S smooth that was virulent and a type 2R rough strain that was non-virulent. Through a series of experiments, it was shown that DNA is the transforming factor. In 1931, female researcher Barbara McClintock began investigating the effects of X-rays on corn chromosomes, which led to her discovery of translocations, inversions, deletions, and ring chromosomes in corn. In 1942, McClintock found the process of transposition in corn chromosomes, and in 1983 she was awarded a Nobel Prize. In 1944, Oswald Avery, Colin McLeod, and Macklin McCarty further proved DNA is the factor that causes transformation. Before then, proteins were believed to carry genetic information, but Avery, McLeod, and McCarty suggested that DNA, rather than protein, may contain the hereditary material of bacteria. In 1946, Rosalind Franklin, a British biophysicist, discovered the structure of DNA. She did this using X-ray diffraction images of DNA that were captured for the first time. In 1953, Watson and Crick used the X-ray data Franklin obtained to confirm the 3D structure of DNA that they had theorized. In the same year, Watson and Crick, as well as a co-worker of Franklin, published an issue in Nature on the structure of DNA. They were later awarded a Nobel Prize for solving the structure of DNA. Unfortunately, Franklin had died at 37 years from ovarian cancer. She was not awarded anything, even though her diffraction images were what confirmed the work of Watson and Crick. In 1990, the U.S. government launched the Human Genome Project. Its purpose was to sequence all 3 billion letters in the human genome, which is the complete set of DNA in the human body. In 1996, Dolly the sheep was cloned, and in 1999, the first human chromosome is decoded. In 2000, the Arabidopsis Genome Initiative was created. The first flowering plant genome was sequenced, and in 2001, the human genome sequence was completed and published. In 2013, DNA Worldwide and Eurofins discovered that identical twins have differences in their genetic makeup. Lastly, bringing this home to NC State University, Dr. Trudy McKay created the Drosophila Genetic Reference Panel, which is a community resource for whole genome association mapping of quantitative traits. Genetics has come very far and research is continuing in the spirit of discovery. Who knows what will happen in the coming years?